Let's start by looking at how smooth muscle responds to the autonomic nervous system. So what I'm going to draw here is some single unit muscle. So we'll have many cells arranged in these layers, and here's the gap junctions linking them. Now, when we talked about how the somatic nervous system controlled skeletal muscle, what you saw was motor neurons coming down and having an axon terminal at a specific spot on a skeletal muscle cell at the neuromuscular junction. Smooth muscle with autonomic axons doesn't work quite the same way. So an autonomic postganglionic axon, instead of coming and having a synapse on one particular place, instead that axon will have periodic swellings in it. So this is a autonomic postganglionic axon. So it's either sympathetic or parasympathetic, doesn't really matter, they would look pretty much the same here. And each of these swellings is what's known as a varicosity. These varicosities have synaptic vesicles with neurotransmitter in them, so that when an action potential propagates along this axon, when it gets to the varicosity, it will cause those vesicles to release their neurotransmitter. And then when it keeps going to the next varicosity, now those vesicles will release their neurotransmitter, and so on. So these varicosities are a little bit like axon terminals, except they're an axon terminal where the axon keeps going on the other end of the terminal, so that it's just releasing neurotransmitter from that spot while the action potential keeps going. That means that here in this muscle, if the receptors for that neurotransmitter are here, then that neurotransmitter will cause a response in this cell and that cell, and it will then, since it's single unit muscle, spread throughout the muscle. So this way, one postganglionic axon in single unit muscle can activate pretty much the entire mass of single unit, although it's not usually just one that does it. And it can spread that activity through the muscle by going down to numerous varicosities rather than just individual axon terminals. Whereas each varicosity is almost how that these are releasing what amounts to a paracrine messenger, something that will diffuse into the nearby environment and activate all nearby cells with receptors on it. In multi-unit muscle, it works similarly, just a little bit different. So now let's make some multi-unit muscle. We'll leave off the gap junctions since it's multi-unit. So in multi-unit muscle, if here is our autonomic axon with some varicosities on it, since these cells are not connected with gap junctions, when this one releases its neurotransmitter, it's going to affect the cells that are right around it. So this one might affect these cells. These are the cells that are very close to those varicosities, but not the others. And since it's multi-unit muscle, as opposed to single unit, what happens to those cells that just got affected isn't going to cause the other cells to respond. So a signal down that autonomic axon will only activate the smooth muscle cells that are close to it. Which means that if I want to be able to activate the other ones, I need more axons. So now we might have a second postganglionic axon with its own varicosities. that one can control a different group of cells that are close to it. So this is almost more like the idea of motor units, that the top axon controls the smooth muscle cells that are close to those varicosities. The bottom one controls the ones that are close to that one. So you could get one set of muscle cells or the other, or both, depending on whether you have signals in one neuron, the second neuron, or both neurons. Hopefully that makes some sense. So there are some small differences in how the autonomic system innervates single unit and multi-unit muscle. In multi-unit muscle, you need to have more axons to control the different parts of the muscle. All right, so that's an example of how the autonomic nervous system works in controlling this. Now, let's take a look at how this might work in terms of the local environment. So local environment control. That would include things like paracrine messengers. And the example I like to use here is of a blood vessel that's supplying a particular tissue area. So in this purple area, you can imagine that we have some amount of tissue. Let's say this is some particular bit of, I don't know, the brain. So the cells in this tissue are getting their oxygen from the extracellular fluid around them. And in turn, the oxygen is coming into that fluid from the blood. So blood is supplying oxygen to the extracellular fluid, and the cells get their oxygen from the extracellular fluid. So I'm going to make one small change here. Okay. So the blood supplies oxygen to the extracellular fluid, and the cells get oxygen from the extracellular fluid. Now, these blood vessels are capillaries. We haven't talked about that yet, but capillaries are tiny blood vessels with very thin walls where oxygen can easily diffuse in and out. This blood vessel is an arteriole. And arterioles have smooth muscle in their walls, which can contract or relax. So our question here is, how much blood do we want to supply to this tissue? Ideally, we want to supply just enough to meet its oxygen demands. So we want to supply an amount of blood at a rate that is carrying just enough oxygen for this tissue. If we're sending more than we need, that's a waste. That oxygen isn't going to help this tissue. And if we're not sending enough, that tissue's starving for oxygen. So ideally, we just want to send just enough. So how are we going to decide how much is just enough? What I'm going to suggest here is that we look at the amount of oxygen in the ECF. So let's say that the cells, actually, here, let me back this up a bit. Let's imagine that the cells are using 10 units of oxygen per second. Those units here don't matter. They're using 10 amounts of oxygen per second. So how much do we want the blood to be bringing? Well, ideally, we want the blood to bring 10 units of oxygen per second. Okay, so if those are matched, then everything is good. And what that tells us is the change in the ECF If we're supplying oxygen to the ECF just as fast as the cells are using it, then the amount of oxygen in the ECF isn't going to change. We're not bringing more than they need, and we're not taking away more they're not taking up more than we're bringing, and we're not bringing more than they're taking. Think of it this way. Imagine you have a pile of M&Ms on your desk, and you're eating 10 M&Ms per minute. If I bring you 10 M&Ms per minute and drop them on your desk, then the size of your pile isn't changing. You're eating them at the same rate that I'm bringing them. That's kind of like the oxygen in the ECF is kind of like the size of your pile of M&Ms. If the blood is bringing 10 oxygen per second to the ECF and the cells are using 10 oxygen per second, then the amount of oxygen in the ECF is not changing. But what if the tissue starts using more oxygen? What if, say, it goes to using 15 oxygen per second? But right at first, sorry, I changed the wrong thing. Let's say the cells start using 15 oxygen per second, but the blood is still only supplying 10 oxygen per second. That means that the ECF is losing oxygen. The cells are burning it faster than I'm supplying it, which means that the amount of oxygen in the tissue in the ECF is decreasing. That's not good. We don't want that level of oxygen to go down. So what we're going to do is have the smooth muscle in this arteriole respond to oxygen. So low oxygen in the ECF causes that smooth muscle to do something. Let's think about what we want it to do. If I want to fix this problem, should I cause that smooth muscle to contract so it narrows the blood vessel, or should I cause it to relax so it dilates the blood vessel? Do I want to bring more blood or less blood to this area? Seems to me I want to bring more, because we're not bringing enough oxygen for that tissue. So the low oxygen in the ECF causes the smooth muscle to relax, which means the vessel dilates. So now my tissue is using 15 oxygen per second, but now my blood vessel dilates, and now it's supplying 15 oxygen per second, which means we're back to no change. We're back to stable. We're supplying as much oxygen as the tissue needs. Does that make sense? So if we set it up so the smooth muscle in this arteriole responds to the local oxygen levels, and when the oxygen levels drop, that muscle dilates, that muscle relaxes, that's going to supply blood to the areas that need it. When that area starts using more oxygen and it starts depleting the amount of oxygen in the ECF, that causes the smooth muscle in the arterioles supplying that area to relax. They dilate and more blood comes to that area, so it supplies more oxygen. It makes the blood vessel respond to the tissue's need by making it responsive to the local environment. Amount of oxygen there is just something about the local environment that can control the smooth muscle. So to summarize that, low oxygen causes the smooth muscle to relax, which causes the vessel to dilate. So what if that tissue then stopped using so much oxygen and went back to 10 per second? Well, now, if we're supplying 15, but we're only burning 10, then now oxygen is building up. We're supplying more oxygen than we need, so it's starting to build up in the tissue. That's also not good, mostly because it's a waste. There's probably other tissues elsewhere which could use that oxygen. So what we want that to do, high O2 causes the muscle to constrict. which will lower the blood supply so it matches the tissue again. So high O2 causes the vessel to constrict, which reduces the blood supply so that I'm not supplying excess oxygen. By having this happen at every individual arteriole that's supplying tissue, you can make sure that each area of the body gets a customized blood supply based on how much oxygen it needs. So that's an example of how smooth muscle can respond to local messenger, the local environment, and even local messenger chemicals. You'll want to know this model. You'll want to be able to explain how smooth muscle in blood vessels responds to tissue needs. Also keep it in mind, because when we get to the respiratory system and talk about how blood vessels in the lungs work, they actually work a little bit differently, because there we're not supplying oxygen, we're picking oxygen up. So think about if there's more oxygen available, so more oxygen in the tissue, would you want that vessel to dilate or constrict if your job is to go pick it up? Think about that. Alrighty, so that's an example of local control. Now we can talk a little. We have mentioned that smooth muscle responds to hormones. So smooth muscle also responds to hormones. So for example, a hormone called angiotensin II causes constriction in blood vessel smooth muscle. Most blood vessel muscle, anyway. On the other hand, epinephrine causes relaxation in some blood vessels. Also in the bronchioles. That's through its action on beta-2 adrenergic receptors, which you've already learned about. Angiotensin II works through angiotensin receptors. So, hormones circulating in the blood can also affect smooth muscle. And finally, a lot of smooth muscle has a certain level of intrinsic control. So, in some single unit muscle, you'll have modified smooth muscle cells, which we call pacemaker cells. These cells generally are set up so that they can change their own membrane potential. They can alter the number of channels that are open to cause their membrane potential to rise or fall, which can then cause what amount to smooth muscle action potentials, which cause that particular mass of single unit muscle to contract when it gets that electrical signal. So these can vary their own membrane potential and cause their own contraction signals. So for example, in the digestive tract, you'll have, in the intestines, you'll have rings of smooth muscle around the intestine, which will have their own pacemaker cells. In this case, they're called interstitial cells of Cajal. And those cells will periodically raise their membrane potential to cause contraction and then lower it. So these rings will go contract, relax, contract, relax, when their pacemaker cells tell them to, without anything else giving them the signal. Now imagine if you have several of those rings near each other. If you time the pacemakers right, you could get a wave of contraction that goes contract, 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 which is peristalsis. That's a wave of contraction that can move things along. Or if you change the timing a little bit, you could have two of them contract alternating, which can then act to kind of slosh and churn food back and forth in the intestines, both of which are things you want to do sometimes. So, if we have those pacemaker cells which cause these things to contract and relax, if we then had something else which can sort of tweak the pacemaker cells and their timing, we could set up patterns of contraction between the single units, which cause that part of the intestine to do useful things. That's a kind of intrinsic control which is then further almost programmed by other parts of the nervous system. In this case, something called the enteric nervous system. But the idea is each individual single unit has its own intrinsic pacemaker cells, and then other cells adjust those pacemaker cells to get parts of the intestine or other areas of the body to contract in different patterns. So that's one example of how intrinsic control can be a part of this. All right, so we've now talked about different ways that smooth muscle can be controlled. Later on, we're going to come back to that. I also wanted to mention a few other things. So what signals can it respond to? A lot of smooth muscle can respond to physical force. That is, if you stretch it, it causes it to contract on its own. This is a little different from skeletal muscle stretch reflexes. We talked about how if you stretch a skeletal muscle, it'll contract, but that's because you stretch it, that's picked up by a sensor, a muscle spindle, which talks to the central nervous system, usually the spinal cord, which then sends a signal down the contraction, the motor neuron causing it to contract. In smooth muscle, that's built into the muscle cell. It doesn't need anything else telling it to do it. You stretch it, a lot of smooth muscle will contract in response to that stretch. So they can often respond to physical force, electrical changes, as well as the other things we talked about, local messengers and hormones, as well as autonomic signals, which kind of fall into that same idea. So there's a wide range of things which can cause smooth muscle cells to respond. Toward the end of this last lecture set, we'll show you a little bit about how that works at a molecular level. But our next step is to talk about how smooth muscle works. We spent a lot of time on skeletal muscle and all the thick and thin filaments and sarcomeres and excitation, contraction, coupling and all that. Smooth muscle is going to work differently. And one way you can know that is that smooth muscle isn't striated. Remember, the striations in skeletal muscle come from the alignment of all of those sarcomeres in the myofibrils. And what that made it so that the whole thing was arranged so that it contracted inward, and the bands got smaller. Smooth muscle doesn't have striations, which tells you that the actin and myosin in smooth muscle is not arranged in the same way, not in those big parallel bundles of myofibrils. So let's talk about how it is arranged.